Hello. In the second part, we're going to have a look at the life cycle of a virus, the second type, which is known as, and it will come up in the slide, the lysogenic cycle. So we've just looked at the lytic cycle and now the lysogenic cycle. Okay, so what's all this about? Well, the good news is this one's easier because the first part is exactly the same as the lytic cycle. So what have we got? We've got a bacteriophage, which is the virus attaching to the host cell. In this case, the host cell is a bacteria. And just like before, the virus injects its DNA into the host cell. If you remember last time, it formed lots of DNA as it replicated, but this time the virus DNA, as you can see here in red, becomes a circle. And next what happens is that the virus DNA seems to fuse with the bacteria DNA, creating a big circle. Now, it can actually be dormant in the bacterium for a long time, and we think HIV, for example, can stay for years with people not realizing that somebody is affected. Now, eventually, some of the virus material triggers a reaction and it starts to copy itself, and it then undergoes the lytic stage, exactly the same as before. Okay, so we need to be able to measure the effect of viruses, which viruses divide very quickly, are fast-moving viruses, and which ones are slower. And we do that with the help of this experiment. So what we do is we take bacterial lawn plates, which is basically agar, a type of jelly that the bacteria grows on and gets all its nutrients, and we take a solution of the virus and we add it over. And after a couple of days of incubating, we have a look at the agar plate, and we are looking for any clear circles, because the clear circles means that the bacteria has died. And why has it died? Because the viruses have invaded the bacteria, and the bacteria have split as they burst because they've become full of viruses. So how do we calculate this? Well, we calculate the area of the circle. The bigger the circle, the more effective the virus is at dividing and ultimately killing the bacteria. So we calculate the area of a circle using pi r squared, which is why for all our biology exams, just like all the other ones in science, we need a calculator. Okay, now I'm not going to ask you to make notes on it, but I am going to ask you to look at these past paper questions. Now there are 5 questions over here, and looking at question 2, it's exactly like the life cycles you have already drawn. So please leave out question 3, but I'd like you to take a couple of minutes and answer questions 1 to 2, 4, and 5, and then we will purple pen it. Okay, how have you done? I hope you've really challenged yourself and tried to use scientific detail. Okay, so let's have a look. So the first question, describe the basic structure of a virus. You need to talk about it having a strand of genetic material and the fact that that strand is surrounded by a protein coat, also known as a capsid. 1 mark for that. And then for 2 marks, explain why viruses cannot replicate outside living cells. You need to talk about they need to use the processes that are happening inside a cell in order to make a new virus. 2 marks there. I hope you're using your purple pen And if you haven't quite got the answer right, pause the video and copy out the correct answer so that you can learn from your mistakes. Okay, question number 4. So you had to calculate 2 areas of circles where the virus had killed the bacteria. For area A, pi r squared, pi and 1.2 squared gives you an answer of 4.52 millimeters squared. Tick that if you got it right. Make sure you've said squared because it's area. And for B, your answer should be 6.16 millimeters squared. Pause and write a correction if you need to. Okay, now the last question. Explain which area contained the viruses that replicated the fastest and killed more bacteria. So I've got to look at my data. I've got 6.16, which is a larger area than 4.52. So area B contained the viruses that replicated the fastest. How do I know? Because a greater area, a greater cross-sectional area of bacteria was killed. Excellent work, Year 10s. Have a good week, stay safe, and take care of yourself. Bye-bye.